In 2015, Karmin and colleagues detected a global collapse in Y-chromosome diversity dating to between 5,000 and 7,000 years ago. Poznik and colleagues confirmed a 1 to 17 effective sex ratio based on more than 1,200 samples, meaning nearly 95% of male lineages vanished, while female lineages remained stable. Natural disasters were excluded as causes. More than 250 Neolithic massacre sites, detailed by Fibiger and colleagues in 2023, reveal recurrent patterns of extreme male-directed violence. Case studies at Talheim and Schletz present consistent demographic and forensic signatures, including mass dismemberment and selective disappearance of reproductive age women. These linked genetic and archaeological anomalies reveal systematic elimination of male competitors on a scale unseen in later history. The consequences are still present in the modern genome, but the definitive mechanism remains unresolved. Was this social collapse, clan warfare, or something else? The evidence begins with the genomic anomaly. Karmin and colleagues at the Estonian Biocenter assembled a global data set of over 120 Y-chromosome sequences representing populations from Africa, Europe, Asia, and the Middle East. Their analysis used whole genome sequencing, covering approximately 3.16 billion base pairs per individual. Applying molecular clock calibration to mutation rates, the team dated a sharp contraction in male lineages to between 5,000 and 7,000 years ago. This event coincided across all surveyed Old World regions. The bottleneck was detected exclusively on the Y-chromosome, inherited paternally, while no parallel reduction appeared in mitochondrial DNA, which is maternally inherited. Karmin's findings, published in 2015, established the temporal window for the collapse and ruled out technical artifacts by cross-validating mutation rates with ancient DNA. The methodology combined high-throughput sequencing with coalescent modeling to reconstruct demographic histories. Poznik and colleagues in 2016 independently confirmed the timing and magnitude of the event by sequencing 1,244 Y chromosomes, reporting a reduction in effective male population size to approximately 1/20th of pre-Neolithic levels. These results were consistent across multiple analytical approaches. The convergence of evidence from two independent teams established a discrete, male-specific genetic bottleneck, measurable in modern populations. The anomaly's global synchronicity and restriction to paternal lines presented a challenge to conventional demographic models, prompting targeted analysis of potential sex-specific causes. Poznik and colleagues at Stanford University published a large-scale replication in Nature Genetics in 2016, sequencing 1,244 Y chromosomes from men across Africa, Europe, Asia, and the Middle East. Their results confirmed a collapse in male effective population size to roughly 1/20th of pre-Neolithic levels during the late Neolithic, mirroring Karmin and colleagues' findings. The study calculated a minimum sex ratio of 1 surviving male lineage for every 17 female lineages at the nadir of the bottleneck. Comparative analysis of mitochondrial DNA from the same populations showed no corresponding reduction in female genetic diversity, ruling out any sex-neutral demographic disaster such as epidemic, famine, or climate event. The bottleneck was restricted to the Y chromosome, indicating a process that selectively affected male reproductive lines. The global scale and synchronicity of the collapse were confirmed by identical timing across Old World populations, with no evidence of regional exceptions in the dataset. Poznik's team used multiple analytical methods, including coalescent simulations and mutation rate calibration, to exclude technical artifacts and reinforce the magnitude of the collapse. The persistence of stable female mitochondrial DNA through the same period further excluded random drift or sampling error as explanations. These findings established the event as a male-specific population-level anomaly, transforming the observation from a statistical pattern into a population-level mystery demanding external explanation. Fibiger and colleagues published in 2023, and they compiled trauma data from 2,300 early Neolithic farmer skeletons across 180 sites in northwestern Europe. Over 10% of these individuals exhibited cranial injuries consistent with violence, including blunt force trauma from stone axes and clubs. Fernandez Crespo and colleagues reanalyzed 338 skeletons from the San Juan Anteportum Latinum rock shelter in Spain, dating from 3,380 BCE to 3000 BCE. Their results showed trauma in 23.1% of individuals, with 97.6% of unhealed injuries found on adolescent and adult males. Across Europe, more than 250 archaeological sites from the late Neolithic contain evidence of organized violence, including mass graves, dismembered remains, and clustered cranial fractures. The prevalence of trauma displays a marked male bias, with injury rates in men frequently double or triple those in women. Statistical surveys indicate that these patterns are not isolated incidents, but reflect a widespread norm. The concentration of perimortem injuries on male skeletons, combined with the absence of corresponding female trauma, suggests systematic targeting. These findings, supported by large sample sizes and consistent reporting standards, establish that lethal violence was both common and sex-selective during the Neolithic, providing a demographic context for the observed Y-chromosome bottleneck. The Talheim death pit, discovered in Baden-Württemberg, Germany, preserves the remains of 34 individuals killed in a single episode around 5000 BCE. Wall and Koenig's 1987 traumatological analysis documented injury patterns. 31 victims suffered lethal blows from the sharp or blunt side of adze tools, while 3 showed evidence of arrow wounds. No skeletons displayed defensive injuries, indicating the attack struck as victims fled or were incapacitated. The demographic profile included 9 adult men, 7 older women, and 16 children. Notably, adolescent and young adult women were absent from the grave. The absence of perifractures or forearm breaks across the assemblage rules out close quarters, combat, or resistance. All wounds were perimortem, with clustered cranial trauma, and no evidence of postmortem mutilation. The attacker's use of adzes, a common Neolithic farming implement, suggests the weapons were repurposed for violence. Forensic detail reveals the systematic nature of the massacre. Blows targeted the back and sides of the head, consistent with ambush or pursuit. The missing cohort of young women has led to hypotheses of abduction or assimilation, as no remains from this group were recovered at the site. Talheim's evidence provides a detailed snapshot of Neolithic violence, illustrating targeted killing and selective survival within a single community. The Schletz massacre, excavated at Aspern Schletz in Lower Austria, documents the aftermath of a raid on a fortified linear band ceramic settlement around 5000 BCE. Teschler Nicola and colleagues reported approximately 200 individuals in a mass grave with skeletal remains showing systematic dismemberment and decapitation. Perimortem trauma includes blunt force cranial fractures and sharp cut marks consistent with adze and axe use. Many skulls were severed from the spine, and long bones were separated from joints, indicating deliberate post-mortem processing. The demographic profile reveals a striking absence of women of reproductive age and adolescence, a pattern also observed at Talheim, but with higher victim counts. The grave contains mostly adult males, older women, and children, while young women and teenagers are missing entirely. In 1997, Teschler Nicola documented evidence of torture, including broken limbs and signs that some victims were immobilized prior to death, such as bound wrists. Gnawing marks on some bones suggest exposure to scavengers, but the overwhelming pattern is one of organized mutilation. The settlement's palisade fortifications failed to prevent the attackers from overwhelming the community and subjecting its inhabitants to extreme violence. Schletz expands the typology of Neolithic conflict, demonstrating that massacre sites could involve not only ambush and execution, but also systematic mutilation and targeted demographic voids. Excavations at Herxheim in southwestern Germany uncovered a settlement occupied by fewer than 50 residents at any one time, based on post-hole counts and domestic debris patterns reported by Bottleneck and colleagues in 2009 in the journal Antiquity. Over approximately 50 years, this small community accumulated the remains of 1,000 to 1,500 individuals in more than 80 ditches surrounding the site. The victim count exceeds the local population by a factor of 20 or more, creating a demographic imbalance that cannot be explained by internal events or ordinary mortality. Isotope analysis of dental enamel, published in subsequent studies, identified non-local origins for many victims, some from as far as 482 kilometers away, indicating repeated influxes from distant regions. The accumulation rate, averaging at least 20 processed individuals per year, far surpassed the reproductive capacity or death rate of a village of this scale. The logistical challenge of transporting, processing, and depositing such a volume of human remains suggests systematic, organized activity over decades. This scale discrepancy raises unresolved questions about the methods, motives, and social structures required to sustain such cycles of violence and ritual at a settlement of minimal, permanent size. Osteological analysis at Herxheim, led by Bottleneck and colleagues in 2009, cataloged standardized butchery signatures across more than 1,000 individuals. Forensic microexamination identified mid-sagittal splits on crania, producing sharp-edged skull cups with cut marks indicating deliberate cleaving while bone was still fresh. Long bones displayed spiral fractures and percussion pits consistent with intentional marrow extraction, and spongy bone elements were reduced through boiling as confirmed by heat-altered surfaces. Bite marks were recorded on metapodials and hand phalanges, with human dental patterns indicating direct consumption of flesh. Bone fragments showed repetitive, patterned cut marks, not random breakage, and tongue removal was inferred from incisions on mandibles. Some skulls were refashioned into cups or decorative objects, with evidence of display on wooden stakes. The uniformity of these modifications, repeated across hundreds of skeletons, rules out scavenger activity or accidental damage. Forensic teams documented the absence of defensive wounds, supporting the conclusion that victims were subdued prior to processing. The resulting cut mark catalog provides a reference for methodical expertise in Neolithic human butchery, distinguishing Herxheim from other massacre sites by the scale and consistency of post-mortem manipulation. Strontium and oxygen isotope analysis of dental enamel, as reported by Bulistan and colleagues in 2009 and in later studies, identified that many individuals processed at Herxheim originated from locations up to 482 kilometers away, crossing multiple ecological zones. Isotopic signatures from teeth mapped victim provenance to regions including the Vosges, the Black Forest, and the Alpine forelands, indicating a logistical reach far beyond the village's immediate surroundings. The diversity of origins, confirmed through distinct strontium ratios, rules out local cyclical violence and demonstrates systematic acquisition of victims from distant communities. This pattern suggests organized raiding or exchange networks capable of transporting captives across hundreds of kilometers. The presence of both mountain and lowland isotopic profiles among the remains further supports the conclusion of deliberate, long-range predation, rather than isolated ritual events. The scale of movement, frequency of deposition, and heterogeneity of ecological backgrounds point to a coordinated process that required planning, surveillance, and sustained intergroup contact over decades. These findings expand the interpretation of Herxheim from a site of local ritual to a regional hub embedded within a broader network of violence and human transfer. Neolithic combatants relied on tools adapted from daily labor rather than specialized weaponry. Archaeological inventories from massacre sites, including Talheim and Schletz, list adzes, stone axes, clubs, spears, bows and arrows, knives, sickles, scrapers, and stone hammers as the primary implements of violence. No evidence of metal armor, shields, or protective gear has been recovered from these contexts. Organic coverings, such as animal skins or woven textiles, provided minimal defense. Forensic analysis by Wall and Koenig in 1987 and by Teschler, Nicola, and colleagues in 1997 documented cranial trauma consistent with blunt adze blows and sharp cuts inflicted by stone axes, with injury morphology matching the available toolkit. Projectile wounds from arrows appear in a minority of cases. For example, 3 victims at Talheim. The absence of defensive wounds, particularly perifractures across multiple sites, supports the conclusion that attacks were overwhelmingly one-sided, exploiting the lack of body protection. Actuarial estimates compiled by Gaylor and Moav in 2007 place average Neolithic life expectancy at birth between 20 and 33 years, with the lowest survival rates for males past adolescence. The combination of unprotected bodies, high-velocity stone tools, and short lifespans created conditions in which lethal violence could rapidly remove entire male cohorts from the gene pool. These mechanical factors clarify how Neolithic societies, despite technological limitations, achieved levels of male mortality sufficient to drive a severe genetic bottleneck. Jean-Pierre Bouquet-Appel's 2002 study in current anthropology quantified the Neolithic demographic transition, showing that the adoption of agriculture caused a rapid population increase across Europe, the Near East, and Asia. Settlement density surged as permanent villages replaced mobile forager camps, concentrating people, grain stores, and livestock in fixed locations. Bouquet-Appel's demographic models charted exponential growth curves, with some regions experiencing a doubling of population within a few centuries of farming's introduction. Economic anthropologists, including Samuel Bowles at the Santa Fe Institute, analyzed how this density shift altered the calculus of conflict. Bowles's battle group model, published in Science, demonstrated that larger resource-rich settlements enabled the formation of organized combat groups exceeding those possible among dispersed hunter-gatherers. The proximity of rival communities, each with accumulated wealth and reproductive age women, increased both the frequency and scale of violent encounters. Statistical analysis of settlement patterns from the late Neolithic reveals clustering at intervals that minimize travel time for raiding parties while maximizing the potential for surprise attacks. Archaeological surveys document a parallel rise in fortifications, with defensive ditches and palisade walls appearing at hundreds of sites between 5,300 and 4,500 BCE. These demographic and economic conditions created the structural preconditions for systematic, organized violence on a scale previously unattainable, providing a macro-level driver for the observed patterns of male-specific mortality and lineage collapse. Two competing models have been advanced to account for the male-specific Y-chromosome bottleneck. Zung, A.W., and Feldman at Stanford University, publishing in Nature Communications in 2018, constructed agent-based simulations of patrilineal clan warfare. Their model used deme-structured populations with typical group sizes ranging from 25 to 50 males, low male migration, and patrilocal residence. Simulated intergroup raids occurred at a frequency parameter of 0.02 per generation, with entire demes subject to extinction via violent conquest. The model tracked Y chromosome diversity over thousands of simulated years, producing a 95% reduction in male lineages and star-shaped phylogenies matching post-Neolithic genetic data. The bottleneck effect was not reproduced under scenarios lacking patrilineal inheritance or intergroup violence. Zung and colleagues reported that only patrilineal warfare scenarios generated the observed magnitude and timing of Y lineage collapse, with high reproductive variance among surviving males. Guion and colleagues in a 2024 preprint proposed an alternative model based on peaceful lineal fission within patrilineal groups. Their simulations set deme sizes between 25 and 50 males, with fission intervals of 4 to 6 generations and reproductive variance of 0.6. In this framework, repeated clan splits and stochastic drift led to the gradual loss of Y lineages without requiring violence. Model outputs showed that drift alone, under these parameters, could eliminate up to 95% of paternal lines over several millennia, replicating the intensity of the observed bottleneck. Comparative analysis indicated that both models fit the genomic data, but only the warfare model aligns with archaeological evidence for organized, male-targeted violence. The debate remains unresolved, with future ancient DNA studies needed to distinguish between demographic drift and conflict-driven lineage collapse. The Y chromosome collapse, identified by Karmin and colleagues in 2015 and by Poznik and colleagues in 2016, persists in the genetic structure of modern populations. Archaeological data from Talheim, Schletz, and Herxheim confirm systematic male-targeted violence during the Neolithic. Current genomic studies continue to trace these prehistoric demographic scars. Interpreting this evidence reframes assumptions about the origins of social order and conflict. Human ancestry remains irrevocably marked by the consequences of ancient selective violence.